All right, welcome to This Explainer. Today, we're diving into the secrets behind one of the absolute most powerful and fundamental techniques in modern biology. If you've ever wondered how scientists actually look at DNA, which, is completely invisible to the naked eye, and somehow make sense of it, you're in exactly the right place. We're talking about gel electrophoresis. It sounds kind of intimidating, for sure, but it's actually a beautifully elegant scientific tool. Okay, let's dive into this.
Here's our roadmap for today. First, the purpose of electrophoresis. Second, the molecular sieve setup. Third, electricity and DNA separation. And finally, interpreting the final bands.
Okay, Section 1, the purpose of electrophoresis.
So picture this. You've got some extracted DNA, but right now, it's just this invisible, confusing biological soup. How on earth do you isolate specific pieces of information from that massive tangle of genetic material? Well, that brings us to our foundational definition. Gel electrophoresis is simply a scientific technique used to analyze and separate DNA fragments based completely on their size. That's the real magic here. It's essentially a brilliantly simple sorting mechanism. You aren't sorting by color or shape, you're taking these microscopic fragments and organizing them strictly by how big or small they are.
Why do we actually do this? Well, it's incredibly practical. We need a way to take really complex, messy DNA samples and bring some order to the chaos. By isolating and separating these molecules, specifically by their size, we're literally taking what looks like a biological black box and transforming it into clear, readable data. It's an absolute game changer, because it allows us to uncover hidden genetic patterns that would otherwise be totally invisible to us.
Moving right along to Section 2, the molecular sieve setup.
Now, what's really interesting here is how we transition from the theory of sorting down to the actual physical lab bench. Picture a pristine, highly controlled scientific setup. You've got a power supply hooked up to a main chamber. Inside that chamber, bathing in a liquid buffer solution, is the star of the show, a translucent gel matrix. And right at one end of that gel, there are these tiny, perfectly formed rectangular indentations called loading wells. The whole thing looks incredibly polished and modern.
And that brings us to a perfect analogy. The gel acts as a molecular sieve. If you were to zoom way in on that smooth, translucent gel, you wouldn't see a solid block. Nope. You'd see this microscopic, sponge-like matrix just packed with tiny pores, tunnels, and little structural hurdles. It is literally a microscopic obstacle course just waiting for the DNA to be dropped in.
Which brings us to Section 3, electricity and DNA separation.
All right, so we have our obstacle course all set up. Now it's time to start the race. Let's walk through how this plays out chronologically. First, imagine hearing a soft, precise beeping as the DNA samples are carefully loaded into those tiny wells at the top of the gel. Step 2, you activate the electrical current. Step 3, the fragments actually begin to migrate through the obstacle course. And finally, Step 4, distinct visual bands begin to form.
Let's zoom in on Step 2 for a second. When you switch on that power supply, imagine a subtle, steady electronic hum filling the room. You're creating an electrical field. Electrons start flowing continuously from the negative electrode at the top, right near the wells, down to the positive electrode at the far end of the gel. And here's the kicker. DNA naturally has a negative electrical charge, so, like repels like. The DNA is instantly repelled by that negative top end and powerfully pulled down toward the positive bottom.
And this brilliantly illustrates exactly how the sorting actually works. As the DNA gets dragged through the gel, the physical size of the fragments completely dictates their speed. The large DNA fragments, they're heavy, they're bulky, they constantly bump into the walls of our molecular sieve and get tangled up in the matrix. Because of that, they move way slower and kind of just stay near the starting line. But the small DNA fragments, they are agile. They slip right through the tiny pores of the sieve, moving much faster and migrating incredibly far down the gel.
Just visualize this separation journey unfolding in real time. The electrical current is the engine pulling everything toward that positive pole, but the gel is doing the sorting. You can almost picture the tiny, agile fragments, maybe glowing as bright little particles, speeding way ahead of the pack, while the larger, heavy-coiled fragments just lag far behind. Over a period of time, this microscopic race perfectly separates what used to be a really messy mixture into highly organized groupings based strictly on size.
Let's move to Section 4, interpreting the final bands, and see how this all builds together.
Okay, the race is over, the current is turned off, and now it's time to read the results. When scientists look at the finished gel, they obviously don't see individual DNA molecules. Actually, scratch that, they couldn't even if they wanted to, they're way too small. Instead, they see DNA bands.
These are distinct, visible solid lines in the gel. But what are they really? Each band represents a massive group, we're talking millions, of DNA fragments that are the exact same size. Because they were all the same size, they navigated the obstacle course at the exact same speed, arrived at this precise distance together, and essentially piled up to create a visible line.
Think of reading a gel kind of like reading a map. Scientists use a marker, which is basically a biological ruler of known sizes, in the very first lane. Then they just read horizontally. If sample A has a band that lines up perfectly with the small marker on our ruler, boom, we know sample A contains small DNA fragments. Looking across, sample B also has a small fragment, but check out the medium size. Sample A has a medium fragment, but sample B doesn't. Instead, sample B has a large fragment. We are quite literally reading a genetic profile visually.
So the crucial point is this: decoding a gel is fundamentally an exercise in pattern matching. By reading the specific band positions horizontally, you figure out the exact sizes of those invisible fragments. And by comparing the vertical columns, you can match different samples against each other, like comparing a suspect's DNA to a crime scene or checking a patient for a specific genetic sequence. This is how scientists identify the patterns and anomalies that lead to really groundbreaking discoveries.
It is honestly mind-blowing how a simple combination of gel and electricity can reveal the hidden blueprints of life. We've taken an invisible soup, sent it through a molecular sieve, and turned it into clear, undeniable visual data. It kind of leaves you wondering, if your own DNA was loaded into those tiny wells and separated out in this gel, what unique genetic story would your bands tell? Thanks so much for joining me to explore this. Keep wondering, keep learning, and I'll catch you in the next explainer.